Dear doctors, welcome to 𝗏𝗏𝗃 H. This is our immunology class and we are discussing the two classes together. So the topics are class 7th and class 8, that is what are the differences between primary immunity and secondary immunity, what are the different interleukins, and of course what are the different functional assays to check for the functional ability of all the cells, functional ability of all the cells, of the T cells, of the B cells, etc. Okay. Yes. So starting with the primary immune response, remember the primary response to any pathogen, to any antigen. For example, there is an antigen that is entering your body for the very first time. So the antigen is going to enter for the very first time. So this is going to be a slow or a sluggish response. This response is going to be a short-lived response and it is mainly mediated by IgM. Whereas secondary one, so if this antigen is going to enter in your body for the second time, now the response is going to be prompt, its response is going to be powerful, it is going to be for a larger duration, prolonged, and it is mediated by IgG. Okay, like that you have to remember. Coming on to the functions of interleukins, so please remember that as you can see, that first and 8, the last ones, interleukin 1 and interleukin 8, they are coming from the macrophages, right? Whereas everything else except 7 is going to come from the T helper cells, okay, everyone from the T helper cells, and please, okay, in fact interleukin 3 is also coming from the T helper cells. Now interleukin 7, they tell you as, coming from stromal cells, so you can remember like S for 7 and S is the stromal cell. Now what are their functions? Let us just quickly understand that 3 is just like this, okay. If you rotate 3, it becomes like this, and very similar to M. So remember that 3 is responsible for myeloid cell development, whereas if you see 7 is just like L, so this is L, that is lymphoid cell development, right? Clear? So what are the different other functions? Let's understand. If you have 1, my dear friends, 1 and T look very similar, so it is responsible for T cell activation, okay, T cell activation, okay. Alright, coming on to the other ones, remember that 4, 5, 6, okay, very important, 4, 5 and 6. So you have a reflex which is called as gag reflex, right? So first G means IgG is going to get converted into what? Into IgE. That's why 4 is responsible for class switching. The other one, 5 is IgA switching, 6 is IgG switching, easy to remember. Then coming on to interleukin 8, so 8 is written like this. So if you cut, this becomes 2C. So it is to tell you that 8 is responsible for chemotaxis. Whereas if you talk about class 2, that is responsible for cell mediated immunity. So imagine in your brains that you have got, okay, 2 cells coming together. So 2 cells means what? That 2 cell mediated immunity. So 2 cell means interleukin 2 and of course cell mediated immunity. So this was about the interleukins. Coming on to the functional assays. Functional assays means these are the tests to determine the functional status of the cell. So these are the tests to determine what? To determine the functional status of the cell. For example, in order to check the B cell function, you will have what? You will have the B cells which produce the immunoglobulins, and of course for B cell functions we do the we do the immunoglobulin assay. Similarly for T cells, as I have told you, that T cells are responsible for production of interleukins and interferons. So we do the interleukin assay and interferon assay. For example, a good example is when you combine the T cells with the sheep RBC, if they are binding, it confirms the T cell function. Okay, it confirms the T cell function because remember the T cell is has a strong affinity for the sheep RBC. So if they are binding, that means it is going to confirm the T cell function. And if the T cells are functioned properly, that means there will be a good proliferative response to the antigen. Okay, so proliferation actually we say that's carried by the T cell. So this is how we have to understand. Then you have got a phagocyte functional assay. So what you do, you have 2 dyes, and this has been asked multiple times as a previous year question. So please remember them. The nitro blue tetrazolium test and dihydrorhodamine assay. Okay, nitro blue. So if you are having the colors like blue, rhodamine means red. So these are both the dye-based test for the phagocyte. So remember, if this is a phagocyte, it is putting a straw and drinking some dyes. Okay. So for the phagocytes, you have the dye-based test like nitro blue tetrazolium test and the dihydrorhodamine assays, right? So yeah, this is all about this video, and I think things are clear and retained in your brains. Thank you so much for watching, and God bless you, my dear friends.